Thank you very much for the introduction, and thanks everyone for attending this seminar. My name is Dustin Frost, and I'm a scientist at Thermo Scientific. My interest is in quantitative proteomics by liquid chromatography, mass spectrometry on Orbitrap platforms, and I have the pleasure of developing our tandem mass tag technology in the Chemistry and Mass Spectrometry Reagents R&D team in Rockford, Illinois. Today, I'll be presenting on how we are currently adapting our EasyPep sample prep tools and TMT reagents for higher sample throughput. We've all come to appreciate TMT for empowering us to accurately quantify proteins and peptides across many samples in a single LC-MS experiment. This multiplexing power has driven the scale of quantitative proteomic studies towards hundreds or even thousands of samples, which is increasingly being facilitated by automated liquid handling platforms. Recognizing this, we've recently been focused on developing products that enable researchers to process more samples in less time using high-throughput automation. I'll begin by introducing our EasyPep sample prep chemistry, the different formats that are suitable for different sample amounts, how TMT integrates into the workflow, and our latest tools for automated EasyPep sample processing. I'll then talk about how we are currently maximizing the throughput potential of TMT through a ready-to-use 96-well plate format of solution-stabilized TMT and TMT Pro reagents that are ideal for automated liquid handling. Before we can get to quantifying proteins or peptides of interest by mass spec analysis, we first need to generate high-quality samples. Traditional protein processing workflows consist of many hands-on steps that stretch over multiple days. Anyone who has prepared dozens to hundreds of samples at the bench wishes they could reduce their hands-on time, and they've likely encountered a lack of reproducibility from one experiment to another or between one person's hands and another's. When preparing many samples for mass spec analysis, sample quality and reproducibility are top priorities. In a generalized workflow, tissue, cell, or biological fluid samples are first collected and proteins are isolated and then extracted by lysis, sonication, or mechanical disruption. Then, disulfide bonds are reduced and alkylated. Proteins may then require cleanup prior to digestion into peptides, and the resulting peptide sample requires cleanup to remove salts and detergents to be made MS ready. The problem with variable sample quality and lack of reproducibility stems from the fact that there is no standard workflow for preparing samples, even for the same sample type. Often, labs have a particular set of homebrew sample prep methods and reagents that differ from other labs. Workflows can be difficult, time-consuming, and tedious, and variability is easily introduced if protein samples are not uniformly extracted, precipitated, reduced, alkylated, or digested. Poor reagent quality can also affect consistency, as well as sample loss and increased chance of mishandling due to the number of steps in the process. Ultimately, these issues can contribute to poor quantitative measurements down the line. To simplify MS sample prep workflows and improve reproducibility, we developed a standardized sample prep method with pre-formulated buffers, MS-grade enzyme mix, and SPE cleanup columns, all of which are supplied in our EasyPep sample prep kits. The optimized method is simple, fast, and convenient, encompassing lysis, reduction and alkylation, digestion, and cleanup to generate MS-ready peptide samples in under 4 hours, a vast improvement over conventional workflows which can take up to 2 days. Shown here is a schematic of the chemistry behind the EasyPep workflow. First, cells are lysed using a lysis buffer containing a universal nuclease which degrades both DNA and RNA. This reduces sample viscosity but also eliminates the need to individually sonicate samples. Next, reduction and alkylation solutions are added to the sample and heated at 95 degrees Celsius for 10 minutes for rapid one-pot blocking of cysteine residues. After cooling the reduced and alkylated sample back down to room temperature, a trypsin lyse protease mix is added to digest the proteins at 37 C for 1 to 3 hours depending on the sample type. After digestion, the samples are transferred to pre-equilibrated spin columns containing a resin for peptide cleanup. Samples are washed with 2 different solutions by centrifugation to remove hydrophilic and hydrophobic contaminants such as salts, lipids, and detergents before elution of the clean peptides for LC-MS analysis. Overall, we've found that this method provides more protein identifications with 30% fewer manipulations and 60% less time compared to conventional workflows. EasyPep accommodates a wide range of sample amounts with the option of mini and maxi spin column kits and a 96 well plate kit. The mini spin column and the 96 well plate kit are suitable for samples in the range of 10 to 100 micrograms, and the Maxi kit handles samples between 500 micrograms to 2 milligrams. All enable efficient and reproducible processing of various sample types for MS analysis, and the three formats have equivalent performance for protein and peptide identifications and miscleavages. The workflow is compatible with common downstream applications including peptide fractionation, phosphopeptide enrichment, and peptide quantitation by both label-free and TMT labeling approaches. TMT reagents can be incorporated into most homebrew sample prep workflows, but require the use of amine-free buffers and a pH of 8 to 8.5 to facilitate complete labeling of peptide N-termini and lysine side chains. Here, I show two ways that TMT labeling can be integrated into the EasyPep workflow. Commonly, TMT reagents are used to label peptides immediately after digestion and before peptide cleanup. This allows samples to be combined for a single cleanup step, but the labeling step could be impacted by interfering contaminants that may be introduced during sample processing. Another option is to label samples after peptide cleanup. This approach provides efficient labeling, but may require the use of a peptide quantification assay to ensure that samples are equally mixed before LC-MS analysis. The efficient lysis, reduction, alkylation, and digestion of the EasyPep workflow generates high-quality peptides which are labeled at high efficiency by TMT reagents. This results in more protein and peptide identifications by LC-MS when compared to traditional sample prep workflows. And the SPE cleanup formats and buffers provided with the kits have been optimized to remove excess, unreacted, and quenched TMT reagents, which also contributes to more protein and peptide identifications. With EasyPep and its integration with TMT reagents, we've made strides towards reducing hands-on time, increasing reproducibility, and improving sample quality. Now, we're turning our attention to improving the scalability and throughput of these tools. To do this, we're introducing new microwell plate formats that are ready for use with multi-channel pipettes, assisted pipetting equipment, and fully automated liquid handling instruments. By performing pipetting tasks in parallel with robotic assistance, further gains can be made with reproducibility, and a significant amount of time is saved when processing large numbers of samples. Often, scaling up the number of samples can be accompanied by scaling down individual sample sizes, particularly with TMT workflows in which samples are pooled prior to LC-MS analysis. In the evolution of our EasyPep technology to minimize hands-on time, improve reproducibility, and increase throughput even further, we developed the Accelerome automated sample prep platform to deliver consistent results for any user. Key features of the system include standardized reagents provided in different kits for either label-free analysis of up to 36 samples, TMT 11-plex analysis of up to 33 samples, or TMT Pro 16-plex analysis of 32 samples. The kits are compatible with various sample types and amounts of 10 to 100 micrograms per sample. The touchscreen and custom software wizard guides the user through the experiment setup. It takes less than 5 minutes to load the system with the user's protein samples and the pre-formulated buffers and reagents and micro SPE cartridges provided in the kits. Once the method is started, the system is hands-off, and the instrument performs the work with no interruptions and no risk of mishandling or operator error, resulting in high-quality samples with consistent results across the entire sample set and from day to day. When Accelerome sample processing is complete, the clean peptide samples are ready to be concentrated in the speed vac and reconstituted for LC-MS analysis on an Orbitrap LC-MS platform. Once acquisition is finished, the experimental design can be exported and then brought into Proteome Discoverer for data processing. We've benchmarked the Accelerome platform against manual EasyPep processing and found it to perform equivalently in terms of protein and peptide IDs, zero miscleavages, and reduction in alkylation efficiency with minimal hands-on time. The software, reagent kits, and automated handling work together to provide extremely efficient TMT labeling, with 99.7% of peptides being labeled with at least one tag. While Accelerome works very well for up to 36 samples, we can easily integrate EasyPep 96 kits into other automated platforms for even greater throughput. Shown here is a Hamilton Microlab Starlet robotic liquid handling system with a custom deck layout for EasyPep processing of plasma and tissue samples in a 96 well plate format. To facilitate the sample cleanup, a positive air pressure module is integrated into the system for washing and elution. The entire workflow efficiently processes 96 samples in just 4.5 hours. Compared to manual handling, Hamilton automation achieves small but meaningful gains in recovery for samples in the 20 to 45 microgram range, and also in consistency of pipetting accuracy. 2 sets of 96 samples were prepared using the EasyPep 96 kit on the Hamilton system on 2 different days, and each sample was analyzed on an Orbitrap Exploris 480 system with Vanquish Neo UHPLC using a 15-minute gradient. The automated sample processing workflow yields very consistent, reproducible results from sample to sample, with excellent CVs for the number of proteins, peptides, and PSMs identified across the entire set of samples. For researchers who are sample limited, such as those doing immunoprecipitation mass spec, formalin-fixed paraffin-embedded tissue analysis, and sorted cell work, we've recently introduced a new EasyPep 96 Micro MS kit that is suitable for samples in the 1 to 10 microgram range. Here, we show that for 1, 5, and 10 microgram HeLa cell samples, the kit achieves equivalent performance for protein and peptide IDs, miscleavages, and reduction in alkylation efficiency. We also assessed well-to-well and plate-to-plate variability for 1 microgram digest samples. The optimized micro kit and protocol achieve excellent reproducibility for protein and peptide IDs, with CVs of less than 2% across the 3 replicates. Likewise, performance with TMT was excellent, particularly for 5 to 10 microgram range, though we do begin to see the challenge of working with dilute sample mixtures in the case of the 1 microgram samples. For such low sample amounts, the tag-to-peptide ratios need to be increased significantly, and sample volume should be reduced for best results. Also point out that for this set of LC-MS acquisitions, the 1 microgram samples use less than half the on-column load for the 5 to 10 microgram samples, but still achieved very good ID numbers. To highlight just one application of integrating EasyPep 96 Micro with TMT Pro 18-plex reagents, we compared formalin-fixed paraffin-embedded tissue samples from lung, colon, and breast tumors versus normal tissues. First, 3 biological replicates of each FFPE tissue were deparaffinized, proteins were extracted using the optimized extraction protocol, and 5 micrograms were processed by the EasyPep 96 Micro MS kit. Next, each sample was labeled with a TMT Pro 18-plex tag, samples were combined equally, and the pooled sample was cleaned up using the SPE plate. A peptide quant assay was performed, and the sample was separated into 8 fractions using high pH reversed phased fractionation. The fractions were then acquired by LC-MS on an Orbitrap Eclipse system, and the data was processed by Proteome Discoverer to identify and quantify proteins. Here we see that the TMT Pro 18-plex quantification was consistent for the 4,000 unique proteins and 26,000 peptides identified across all samples, and principal component analysis grouped the biological replicates based on similar protein and peptide abundance. We were then able to identify unique protein abundances for a few putative protein biomarkers across the 3 conditions. ERBB2 receptor tyrosine protein kinase showed a 6-fold increase in breast tumor compared to breast normal samples. This protein plays an important role in the development and progression of certain aggressive types of breast cancer. CECM5 showed a 2-fold increase in colon tumor compared to colon normal samples. This protein plays a significant role in the inhibition of apoptosis and differentiation in colon cells. And coronin 1A showed a 2.5-fold increase in lung tumor compared to lung normal samples. Now we'll focus a bit more on realizing greater time savings and throughput with TMT multiplexing. It's easy to see how sample multiplexing increases throughput by allowing parallel analysis of many samples in a single LC-MS run. For instance, analyzing 18 samples with 3 replicates each using a 3-hour acquisition time equals 1 week of non-stop instrument time. By labeling with TMT Pro 18-plex reagents, the pooled replicates can be analyzed in just 9 hours of instrument time. This method also enables fractionation of the combined peptide sample to achieve similar depths of protein coverage as label-free analyses in less than 1 day. Performing a single LC-MS analysis of different samples from independent experiments also increases the overlap among replicates, leading to fewer missing protein identifications and quantitative values. And when sample limited, combining multiple samples into one boosts MS signal levels for more confident protein and peptide identification. Despite advances in mass spec instrumentation, there can be significant missing values when samples are run independently. This is due to large dynamic range of protein abundance in samples that interfere with low abundant protein detection and the stochastic nature of the acquisition methods used where precursors are selected by intensity, retention time, and mass, but not by sequence. Therefore, in large data sets, only the proteins which are identified in every analysis can be confidently used for quantification. To demonstrate this, 30 HeLa cell digests were prepared and individually labeled using either 3 sets of TMT 11-plex reagents with a common bridge channel or with 2 sets of TMT Pro 16-plex reagents with a common bridge. Since each multiplex set is acquired separately, we would expect more overlap in the samples with the fewest number of runs. On the right are Venn diagrams showing the results of the experiment. For the 11-plex TMT runs, nearly 2,900 proteins were quantified across the 30 samples, whereas over 3,100 proteins were quantified across the 2 16-plex TMT Pro runs. This represents a 10% increase in quantified proteins just from fewer missing values among replicates. So now that we understand how multiplexing benefits us, how do we employ multiplex TMT reagents in a high-throughput manner? By offering TMT reagents in 96 well plates. We've already seen users reformat bulk reagents supplied in tubes into 96 well plates for labeling large sample sets with the aid of liquid handling platforms. Now, we're gearing up to launch a ready-to-use 96 well plate format containing multiple sets of TMT 10-plex, TMT 11-plex, TMT Pro 16-plex, and TMT Pro 18-plex reagents in each plate. Reagents are pre-aliquoted into each well in 5 microliters of stabilized dimethyl sulfoxide solution in amounts of 40 micrograms TMT and 50 micrograms TMT Pro, which is suitable for labeling 1 to 10 micrograms of protein digest. DMSO was chosen over acetonitrile due to its moderate viscosity and non-volatility, both of which are critical for success with robotic pipetters and automated workflows that process many samples over many hours. Already, we've seen users opt for DMSO over acetonitrile when using liquid handlers to facilitate TMT labeling. The plates will be sealed with a peelable, pierceable foil, and each plate is barcoded on 2 edges with a unique identifier. Here is what the ready-to-use reagent plates will look like. The label on the pouch will show the reagent layout with some brief instructions for use. First, equilibrate in the pouch to room temperature, then centrifuge the plate to gather the reagent solution to the bottom of the wells. Then, peel or puncture the foil seal to access the reagents. Labeling is performed as we are all accustomed to, by adding 1 to 10 micrograms of protein digest and 10 microliters of 100 millimolar TEEB or HEPES buffer to each well and incubating for 1 hour. Then quench, combine, and clean up directly using our EasyPep SPE formats, or dilute the DMSO concentration to less than 5% and proceed with reversed phase cleanup or fractionation. Since the reagents are pre-formulated in the DMSO-based stabilization solution, we've benchmarked performance against reagents in acetonitrile that users are accustomed to. Here we show that both solvents perform comparably in terms of protein and peptide ID numbers and labeling efficiency. We've also evaluated the reagent stability over time in the DMSO-based stabilization solution in the 96 well plate. Experienced users of TMT are familiar with how short the shelf life of the reagents can be once the pouch is opened, or when the reagents are reconstituted in acetonitrile. With these new solution-stabilized reagents, we achieve far greater stability. Here I show the results for several stability time course experiments conducted over many days or months under different storage conditions. At each time point, reagent solutions in the 96 well plate were unsealed and analyzed by direct infusion mass spec to measure the peak intensity of the active tag versus the inactive hydrolyzed tag to determine the percent of the active tag. Note that since reagents are added to peptides in excess during labeling, some loss of activity is acceptable and complete labeling can still be achieved. At room temperature, we see that the sealed reagents retain much of their reactivity even after several weeks. In the fridge, the reagents remain viable for several months, and in the freezer, we would expect between 9 to 12 months of stability, if not longer. Additionally, the reagents are tolerant of many freeze-thaw cycles. Since automated workflows of many samples can often take a full day to complete, we also assess the stability of the reagents exposed to air in an unsealed plate. Here we see that reagents on the deck of an instrument can retain their activity for a full 8 hours. Even if the instrument were to encounter a fault and the process had to be resumed the next day, the reagents on deck would still retain enough activity for a successful labeling step. We're impressed at how much more stable these reagents are in this DMSO-based stabilization solution, and we think our users who process hundreds to thousands of samples a week using liquid handling equipment will appreciate this as well. So to wrap things up, we've shown that our EasyPep sample prep kits produce high-quality samples for both label-free and TMT-based quantitative workflows at various sample scales and for hands-on work or by automation. We've also highlighted the Accelerome automated sample prep platform that allows push-button processing of over 30 samples at high reproducibility with minimal hands-on time. And now we have new EasyPep Micro and TMT 96 well plate formats that are especially well suited for high throughput processing of hundreds of samples on fully automated platforms. I want to recognize and thank my many colleagues at Thermo Fisher Scientific and at the UAC Rockford Medical School who contributed to the development of the technologies and solutions that I presented today. Thank you all for attending, and I look forward to the roundtable session at the end of the day.